afternoon, or good evening, depending on where you are. Welcome and thanks for attending today's Microfluidics webinar titled Solvent-free Production Method for Liposomes and Lipid Nanoparticles. My name is Yang Su and I'm the Manager of New Application and New Technology and Applications at Microfluidics. I will serve as your moderator for today's webinar. Before I introduce you today's speakers, I'd like to quickly go through a few housekeeping rules. This is a live webinar, and as I have mentioned already, we have started the recording already, so I will ask everyone please remain muted. Also, please turn off your webcam to ensure the webinar goes smoothly without any interruption or lagging. We will take all questions at the end. There will be a Q&A session after the presentation. Please submit your questions via the chat window. We will try to answer as many questions as possible. If we don't have time to go through all of them, we'll certainly follow up with you after the webinar. The recording of today's webinar will also be available and accessible via our website shortly. As I've mentioned, we have a very interesting topic today, which is producing liposomes and the lipid nanoparticles without using any solvent. I've introduced myself already. Now let me introduce you today's speakers. We have two speakers today. Our first speaker is Jaquin. Chris is currently the applications engineer at Microfluidics and has amassed the knowledge of both microfluidizer technology and applying it to a wide range of applications, including various lipid-based nanoparticles, through closely working with customers in numerous proof-of-concept tests, process optimizations, and scale-up tests. Our second speaker is Dr. Yvonne Perry. Yvonne is currently professor in drug delivery within the Strathclyde Institute of Pharmacy and Biomedical Science at the University of Strathclyde. She is an internationally recognized expert in liposome and particulate drug and vaccine delivery research. Her multidisciplinary research has been recognized by a range of fellowship and awards. She currently serves as president of the Controlled Release Society, as well as chief and associate editor for several high-impact journals. Without further ado, I'm going to hand it over to Chris first.
Yep, thanks, Yang. I'll begin today by quickly going over the agenda for today. I will be starting by giving a brief overview of Microfluidics, discussing who we are as a company, a very high-level overview of some applications, along with how our technology works and our product line. At this point, I will be handing off to Dr. Perry to discuss the solvent-free production method, specifically touching on nucleic acid therapies, aqueous-soluble small molecules, and lipid-soluble drugs. At the end of the presentation, as Yang had previously mentioned, we will have a brief Q&A session. So who is Microfluidics? Well, Microfluidics was founded in 1982, and we've been producing high-shear fluid processors utilizing our proprietary fixed geometry interaction chamber technology ever since. We are headquartered just outside of Boston, Massachusetts, but we have localized support internationally. Sold over 4,000 processors to over 2,000 companies. Back in 2011, we were acquired by the IDEX Corporation and have since been grouped with our sister companies Quadro Engineering, Fitzpatrick, Matcon, and Cerados to form the MPT, or Material Processing Technologies Platform. Our processors can be used for both R&D and manufacturing for a variety of materials across a variety of examples, inkjet inks, coatings, and various nutraceutical and cosmetics applications. We have a vast applications and machine design experience, and our team is always here to help support you. So these are some common applications that we see our technology utilized for. For nanoemulsions, liposomes, polymer nanoparticles, and other similar droplet-based systems, our technology ensures that you are able to create very small and uniform droplet-sized distributions. And for these particular applications, we are commonly able to be sterile filtered as well. We are also a very efficient method for cell disruption, and our customers typically see very high levels of protein recovery. Similarly, some other common applications used in our technology are to reduce the molecular weight of polysaccharides, deagglomerate particles, along with reducing the particle size. Similarly to the droplet-based systems, we are able to create very uniform small distributions of these systems as well. So how does microfluidizer technology work? You would load your product into the inlet reservoir, where it is then pulled via the constant pressure intensifier pump through our fixed geometry interaction chamber at pressures of up to 30,000 psi, where your material will be subjected to constant high shear rates and impact forces. As it is a fixed geometry interaction chamber, all of your material will be processed under the exact same conditions. After passing through the fixed geometry interaction chamber, your product will pass changer or a cooling coil in order to control the temperature, and at this point your product will be collected. We offer two separate styles of interaction chamber, both the Y type as shown on the figure above and on the Z type as shown in the bottom figure. For the Y type interaction chamber, your material would actually separate into two separate streams and pass through one of these two small microchannels. After passing through the microchannels, your product would actually impinge upon itself in the high impact zone, and this can lead to very intensive micromixing. The Y-style interaction chamber is typically used for liquid-liquid dispersions, so as I mentioned, potentially nanoemulsions, liposomes, and polymer nanoparticles. The other style interaction chamber that we offer is the Z-type interaction chamber. As can be shown in the diagram below, there is only one microchannel for the Z-style interaction chamber, and after exiting the interaction chamber, your product will face high impact forces up against this wall. The Z-style chamber is more commonly used containing solids, so various suspensions, dispersions, and cell disruption as well. So one additional benefit of microfluidizer technology is that we can guarantee scalability. So on the left-hand side, you can see the schematics shown on the previous slide of both a Y-style and a Z-style interaction chamber that might be on a laboratory or a pilot scale piece of equipment. What we do in order to ensure scale-up is we simply place these microchannels in parallel, which allows for a higher throughput of your material. The fact that we use the same dimensions and the same geometry for these microchannels ensures that your product will face the same uniform, consistent processing conditions, whether you're processing on the laboratory or the production scale, and thus ensuring you'll be able to achieve the same results, assuming the rest of your processing parameters are also the same. So that brings me to my final slide, just briefly to show you our product portfolio. As can be shown, we offer a variety of lab scale, pilot scale, and production scale processors capable of handling batch sizes as small as 1 milliliter on our smallest lab scale units, up to thousands of liters for our biggest production scale units. Additionally, all of the processors that are shown with these dots on them can be sold with cGMP capabilities. If you have any additional questions regarding our technology or our product portfolio, please feel free to ask it in the chat window or send me an email. But at this point, I will be turning over the presentation to Dr. Perry.
I will now share my screen, hopefully, if you could just confirm once I get it up. Oops. Okay, is that on slide presentation mode?
Yes.
Yeah, we can see.
Thanks. Thank you. You never know, so it's always good to check. Okay, thanks for joining. And yes, we're going to be talking about the work we've been doing here at the University of Strathclyde on solvent-free manufacturing for both liposomes and lipid nanoparticles. So in general, we know the advantages of drug delivery using lipid-based delivery systems. So we have liposomes, we have our lipid nanoparticles, which we're hearing a lot about on the news, obviously, through the COVID vaccines. But we have to remember the role liposomes also play in therapeutic reasons for drug delivery. We also have solid lipid nanoparticles, polymeric nanoparticles, and microemulsions, all which are in the nanoparticle range and can improve drug delivery and efficacy. So about a year ago, nanomedicines, as you know, were very much still a research tool. We had lots of evidence about them working in the lab. Scalability and manufacture was a bit of a valley of death for us, and that was causing problems for us to get to market. Now, we are seeing people tackling that very rapidly to try and help the vaccine element of what we're facing at the moment, but we have seen problems even in previous production issues. So back in April 2013, we had a shortage of doxorubicin encapsulated within liposomes, known as calyx in the UK, but doxil in the US, because of manufacturing issues. So it is a problem that we needed to tackle, and actually still we are tackling it, and you're seeing that coming through in the news. One of our research aims within the university was to develop a solvent-free manufacturing process for lipid-based drug delivery systems. And really, there's several drivers for that. Obviously, there's issues around handling solvents in terms of safety. Also, we want to be leaner and greener in our manufacturing processes, and the additional impact that has on finance and time and accessibility to these products. So we've been working on that. The process we've been developing is basically various steps where we can hydrate the lipids straight into an aqueous solution, but they do form very large particles, so we then reduce the size using homogenization as shown here on the screen, and then we can do buffer exchange, purification, concentration to produce product at the concentration required, and also we can sterilize it then. So this is our basic production pathway we were looking at. So I've basically now developed 3 case studies to go through this for us, and I'll start with nucleic acid therapies because I know this is the hottest topic we're all talking about. Who would have thought? So the journey of nucleic acid. So it's not that new. So back in the 90s, we had John Wolfe who had his first paper where he injected muscle, and we did see a response. That didn't translate into products because the immune response wasn't very strong. But building on that, then the field moved to RNA, and we now know we've got synthetic RNA, or we've got the self-amplifying RNA, and both of those are being looked at closely now, coming through in the products in clinical trials at the moment for the vaccine. And they have various advantages. The self-amplifying RNA will require less doses. But in each case, you have your RNA, encodes for the protein antigen. This then creates the immune response within the body. Now, RNA in general, like DNA, is highly anionic and hydrophilic. So that means it's very difficult to get into the cell membrane. So cellular uptake is a problem. It's also quickly broken down by RNAs. Anybody that works in it knows that's a challenge both in the lab and in vivo. So we need a delivery system, and all the products we're seeing being tested in clinical trials are all using lipid-based delivery systems at the moment. So to deliver RNA, we've been looking at liposomes, LNPs. We've also looked at solid lipid nanoparticles, which are like polymeric nanoparticles, but based on a lipid. Also cationic microemulsions. And we've actually been looking at all of those systems. And it's not to forget there is a product already approved for use on Patra, which uses an ionizable lipid nanoparticle, which has been used not for a vaccine, but for therapy. So we wanted to first look at, when we started looking at RNA vaccines, so this was a project funded by Horizon 2020, and we started it 3 years ago and finished it in January past, at the start of the year. And we actually compared liposomes, solid lipid nanoparticles, polymeric nanoparticles, and emulsions. And we looked at putting the RNA on the outside or on the inside, and we used various methods at lab scale to look at that. And we also tested 2 different cationic lipids, and we then tested them. So the 2 cationic lipids we tested was dioleo trimethylammonium propane, or DOTAP as it's more commonly known, or dimethyl diammonium bromide, or DDA as it's more commonly known. So you can see they both have a cationic head group. And we've looked at cell uptake, immune responses. So we did an early screening program for physical chemical attributes, and there was a lot of data, and it's been summarized into this slide, which is unfortunate because it was so much work, but you can basically see we were able to produce nanoparticles around about 100 to 200 nanometers in size, depending on what the base formulation was. So the liposomes were a bit smaller, around about 100 nanometers. Our solid lipid nanoparticles increased in size to about 150. Our polymeric nanoparticles were around 200 nanometers, and our emulsions were about 175 nanometers. All carried a positive charge, all had high loading because of that positive charge. So you've got an anionic RNA complex into a cationic formulation. And we did test them for RNA's degradation, how much they offer protection, and they all gave high protection by condensing the RNA and blocking breakdown by the enzyme. So that was great. So we had them. So we wanted to then progress these in vitro screening. So we basically fluorescently labelled our particles with a Dill stain, so we could measure the fluorescence. We then took them into cell lines and we tested toxicity, cell uptake, and expression using a luciferase encoding RNA. So here we can see, so we compared the two cationic lipids. So we haven't moved on to ionizable lipid yet, because at the start of this project, they weren't very easy to get a hold of back in 2000, this time last year. So we've got the cationic lipid DOTAP or DDA. And we looked at cell viability. So generally DOTAP is less toxic than DDA. DDA is a bit more toxic. We then looked at cell uptake, which is measured in fluorescent intensity. And basically what you can see here, so here we have liposomes with the RNA inside, outside. We have the solid lipid nanoparticles with RNA inside and outside. For DOTAP, we only have PLGA nanoparticles with the RNA inside because we couldn't formulate the ones outside it precipitated. And for emulsions, we always have the RNA in the outside because you can't put it in the oil phase. So you can see from fluorescence, there is some differences in cell uptake, but nothing dramatic. If you look at the DDA, definitely the polymeric system shows a much higher cell uptake. But when you translate that into transfection, you actually see no notable difference between the formulations. So the uptake didn't translate into differences in transfection in the cell line. So there was no notable difference between these formulations in that respect. So it didn't matter the cationic lipid used, and it didn't matter the formulation base. Was it a nanoparticle, a lipid nanoparticle, or an emulsion? But we did have to down select in some way. So we then took forward two different liposome-based systems, or LNP-based, depending how you want to define it. So we had DOPE, a fusogenic lipid, combined with DOTAP, or DDA, and we put the RNA inside. And we also tested PLGA DOTAP nanoparticles. So they're made with PLGA and DOTAP, and we also again put the RNA in the inside. So we did an in vivo study with 1.5 and 0.15 micrograms of self-amplifying RNA encoding for a rabies virus. This was because we have a benchmark vaccine commercially available for that we could compare to. We gave doses at 0 and 28 days, and we tested both cellular and antibody responses. Here you can see the rabies anti-IgG responses at two different doses, 1.5 and 0.15, with DOTAP nanoparticles, DOTAP lipid nanoparticles, or DDA nanoparticles. And then we also have a cationic emulsion that we added into this, so that also has DOTAP in the formulation. And then the last one, Rabipur, shown here is a commercial product. It's not RNA based, but it is the one that's used in humans. So it's given to mice at a 1/20th of the dose. And then on the graph, we have the IgG. The blue dashed line is below the limit of detection, and the red dashed line is where we get protection. So any level above that would be seen as an effective vaccine. So what you can see from this graph is that all three candidates, compared to Rabipur, gave us a response above the protection limit. So particularly at the 1.5, we had no problems. The DDA was a bit more variable, but certainly the DOTAP nanoparticles, LNPs, and certainly the emulsion all gave us very good responses. We then broke it down into the CD4 and CD8 responses. So I appreciate there's lots of colour and lots of data here, but you're really just looking for the biggest columns. So we see our DOTAP nanoparticles working well and our cationic emulsions working well and equivalent to the commercial benchmark product. So here we have CD4, and this is all the various types of cytokine responses. And here we have CD8 again. You can see that the DOTAP nanoparticles giving us equivalent responses to the commercial product Rabipur. at this stage. And it was a benchmark just to make sure we could get how we could compare our products. So although there was no significant difference, the nanoparticles at 1.5 micrograms of SAM certainly induced higher frequencies of CD4 T cells compared to the other formulations. And we see a similar profile for the CD8. So that was very encouraging that we could produce these immune responses with a SAM vaccine. But our liposomes were weak, and we thought, well, that's interesting, let's look at that more. So we did then compare a panel of cationic lipids here. So we've already talked about DOTAP and DDA. We looked at another panel of these cationic lipids that were commercially available. And the idea was for us, it was commercially available. There was no IP associated with it, so it was open source for us. And we basically had two combinations where we had either the DOP and the cationic with PEG, or we added an additional lipid, distereophosphatidylcholine, as a stabilizing lipid. And that's the one you're kind of seeing in most of the LNPs at the moment. So we did a big screening program, and I appreciate this is a lot of data, but really what I'm trying to show here is the red box ones. So we took an LNP-based formulation where you have DSPC, cholesterol, cationic lipid, and PEG, and the PEG's on the surface to help stabilize the system. And the cationic lipids we tested again were DOTAP and DDA. So you can see the formulations are about 100 nanometers in size, PDI about 0.2. They're neutral in charge now because they are pegylated, but they do still complex the RNA and put them inside. So DSTAP, which is a high transition temperature lipid, just to point out, was very difficult to work with and produced high aggregates. And then we have the ionizable lipid, which is the one that was becoming commercially available, which is DLN-DLA, which basically you take the pH down and it's cationic, bring your pH up and it becomes neutral in charge. That allows you to capsulate and then changes charge. And that's shown here by its zeta potential. You can see he's almost neutral. So again, we took them into the exact same vaccine protocol study. Again, we have our Rabipur as a benchmark at 1/20th of the dose. So we have our DOTAP LNPs, our DDA LNPs, a cationic emulsion that we brought forward again because that was a very effective formulation, and the ionizable lipid with the DLN-DLNA, which is a commercially available ionizable lipid. And what you can see is really there's no significant difference across most of the platforms between a cationic system and an ionizable lipid, which is much more expensive compared to these cheaper cationic formulations. And if you took it 2 weeks later after the second injections, again, for this would be us at 6 weeks after first vaccination, all the responses have gone up 20-fold. You can see that the profile is strong for all formulations. So that really excited us that a 1.5 microgram dose of RNA, self-amplifying RNA, was equivalent and a very cheap and easy to produce in our labs because the lipid is much more accessible, equivalent to the IP-controlled DLN and a lot of the ionizable lipids. So it was a very good to see that you can create immune responses through these processes. And again, just to say, we also have T-cell responses. So again, this is CD8 responses broken down by your immune profile. But generally, you're looking for the highest bars, and you get CD4 here. So with CD8 cells, we mainly get a Th1 type response, characterized by interferon gamma alone or in combination with TNF alpha and IL-2. So basically, you can see that's the blues mainly. And DOTAP and DDA gave similar profiles, and the ionizable LNPs did give a significantly higher frequency, and that is shown here. You can see a higher response. With the CD4, there was no significant difference unless you used the higher dose of the ionizable lipid in the formulation. So very good responses all round. We're talking very low doses here, which is also excellent to see. So that's great. So we have that. How are we going to scale that up to solvent-free? So we've been working on this now, and we're now taking this forward. So we've been looking at DOPE and DOTAP, the initial transfection formulation that if you worked in the field of gene therapy, you'll know very well. So we take that in a 1-to-1 combination. Again, we hydrate and homogenize. Now we're being careful in keeping it at room temperature to avoid breakdown. We We're then passing it through, and then we added the DNA. So this is a post-loading, and the advantages that could bring us, you could have mass production of the transfection agent or the vaccine agent, which you can have going alongside the production of the RNA. So it could, half the time, rather than having it all at once, you have two running in parallel and then combine at the end. So what we have here is the data where we have the negative to positive ratio. Before we add in the plasmid DNA, the liposomes are around about 50 nanometers. They do go up in size as they aggregate and restructure, and they go up to about 100 nanometers. They generally keep their positive charge, and because of positive charge, we get excellent DNA loading. So that will be in a configuration that we will explore more. But in terms of efficacy, so we then move to cell culture studies, and we look at luciferase expression again, and you can see the ratio of 4, basically negative to positive, gave us the best luciferase immune responses, so the best transfection levels. Sometimes in some of the other studies, a ratio of 6 is better, so it's generally 4 to 6 are the chosen ratios. So that was good to see. We then wanted to see, well, if you don't produce them in parallel and you want to in situ load as an all-in-one process, can we do that? So we're currently looking at that at the moment. So in that case, you add your powdered lipid, and your aqueous buffer then has your, in our case, DNA or your samRNA, mix, hydrate, and reduce size to the 110P that we are using. And again, you can see you can bring your size down to about 100 nanometers with PDI of below 0.3, the positive charge, and our loading is still very high. So that's with the DOP DOTAP. Then when we use the formulation with DSPC in it, which is more akin to the lipid nanoparticles we're seeing coming through in vaccine studies, we can use the same model, hydrate with the RNA, we can get sizes down to about 200 nanometers, we keep a positive charge, and our loading is very high. So we're taking these forward now into transfection and hopefully in vivo studies very soon. But one thing we also looked at, and it's a very hot or very cold topic, I guess, the problem with the cold chain supply. So the fact that some of our products will have to be stored at -80 for the vaccine. So we've started looking at that now and figuring out if we can just use cryoprotectants to preserve these systems. So this is the formulation. And I should say Valerie in our lab has been doing a great job with this. So we have this is the formulation made. So it's got the DNA loaded inside. We then freeze it at -20 with no cryo. We put sucrose at 10% or trehalose at 10%. And we're seeing the sucrose is retaining the physical attributes in terms of size, zeta potential, and DNA loading. Now, obviously, the next step is to check how these work in vivo, and they will progress to that. But it's exciting to see at least frozen at -20. They are staying stable in that respect. So that's the LNPs and the vaccines, but we can't forget that we still need delivery for small molecules, and there's a lot of work to be done in that. And just acknowledge again the key players in doing this in our lab were Swapna, Carla, Rachel, and Valeria. So all have been working really hard on this. So again, the production of liposomes, we have an aqueous core, and we can put the drug in the aqueous core or the bilayer. And again, we're using manufacturing. We're using the M110P, we purify by TFF. If we're loading the drug by pH gradient, I'll explain we can load it, or you load it at the beginning, sterile, and then you've got your product. So the first product we looked at was a pegylated liposome incorporating doxorubicin. You might call it a doxal mimic. It uses a similar uses the same formulation parameters. So we have 3 lipids, HSPC, cholesterol, pegylated lipid, and the drug. So again, we hydrate our lipid, we homogenize it down to size, we produce liposomes. We can then change the buffer externally to create a pH gradient, and we can remote load the drug. So first stage is making the small unilamellar vesicles. So here we have 3 different pressures, 15, 18, and 20,000 psi. The pass is how many times we put them through the system, and the particle size. So obviously before they've been downsized, they're very large, they're about 1,500 nanometers, if not more. PDI is huge, so that's why I'm saying the particle particle size you can't really trust given the PDI. But it gives you an indication these are very large particles. And you can see the intensity is very large combination of sizes. But after just one pass, you can see we're starting to get our SUVs in a narrower product. We can 2 passes and then 3 passes. We're down at 100 nanometers with a low PDI. And that's at 15,000 PSI. We took it to 18,000 and then you can see 20,000. We're really bringing the size down, and we like the 20,000 PSI. The lipids were happy, they could handle that, no problem. We have a nice PDI below 0.2, zeta potentials around about neutral as we would expect. So these don't have a pH gradient yet, they're just formed liposomes. We then purify them using TFF, tangential flow filtration. So just basically cross-flow filtration where the liposomes go through and we can wash the external media away, so we have a different pH gradient. This is just us showing you that how we change the outside buffer, and normally 12 ml or 6 washes for us was able to wash our liposomes and create this difference with 2 different pHs inside and outside. And that's probably explained better here. So we create a pH gradient so that when you add the doxorubicin on the outside, it transfers into the liposome due to the difference in pH. The drug crystallizes and becomes entrapped. And that can actually be seen here in this. These are 2 cryo-TEMs, so these are liposomes empty with a pH gradient. We then load the doxorubicin, and you can see the lines, so that's it crystallizing out. And the liposomes are actually red because doxorubicin inside them is red. In terms of loading time, so you heat up your liposomes, you take them to 60 degrees so that the bilayer becomes fluid and the drug can cross the membrane. And you can see that at 60 degrees, we can do loading within 30 minutes of that. So that was great to see. We also did test 2 different lipids, hydrogenated and phosphatidylcholine, just to check the process. Loading is always high, and as I showed you, I've zoomed in this picture to make it bigger. You can see the crystallized out liposomes in the product, very similar to what you would see with dox. So that looked really good, so we're very happy with that. The size was in range, so we're trying to create a doxal mimic, so the size was in range, nice low PDI. So just mapping, we did test the process. So this is us doing it on the bench scale with solvent. We got decent loading, decent size, although slightly higher. We probably could have improved that with further sonication, but we weren't taking it in vivo. But good loading, but you can see when we do it with the solvent-free method, we get very high drug loading, very nice particle size, and it's a scalable, robust method. So that we were very happy with that. We then wanted to test the other attributes. So drug release is very important because it affects the PK profile, the pharmacokinetic profile and targeting of the liposomes. So with pegylated doxorubicin, we want it to go to the tumour site, stay there, release the drug and kill the tumour cells. So USP4 is just a discriminatory model where we're trying to look at release profiles and tease out if there's any difference in it. And there's been really nice work done by Frank Zoka and his team working with FDA to show differences in some of the generic liposome products. So this is us. So we did it over 6 hours. We have elevated the temperature. So we're not trying to mimic an in vivo environment. The temperature is up at 45 degrees. We're really trying to stress the liposomes to see if they release at the same rate. So we're not trying to simulate in vivo conditions. So free drug comes out of the system very quickly, as you would expect. But in terms of our liposome products, didn't matter if we were past 2 or past 3 or pressures 18 to 20, we got the similar product in terms of release profiles. So that was great to see, and it shows you've got some latitude in your process conditions when making this product. We also did look at a non-pegylated liposome. So not only is it not got PEG in it, it actually uses a lower transition temperature lipid, so egg phosphatidylcholine, and it uses a different buffer for us to look at the pH loading method. So exact same process, we produce the liposomes, we did buffer exchange, we load them, and then we sterilize them by 0.22 filtration. Particle size is retained, PDI retained 0.2, very low. You can see the profiles here. We then look at drug loading. Obviously, there's no drug before we put it in. Then at drug loading, we have high, and after sterilisation, it is also high. And here's the liposomes. Here you can see, so that with the different buffer gradient, you tend to get a slightly less rigid doxorubicin crystal, so it's a slightly different construct to see inside, similar to the product we were copying as a commercial product. That's great. So we've put LNPs and we put RNA, we put water-soluble drugs in the core. The other thing liposomes are great for are dissolving lipid-soluble drugs. So again, the team Swapno, Carla, Rachel and Valeria looked at this for us. So now what we do is we're hydrating the lipid and the drug at the same time. Homogenize it down, reduce the particle size and control that to what we want to use. So our protocols are here. So we're using a succinate buffer. We do form a complex and then add in our lipids, and we're working at 65 degrees here, 65 to 70. And we took a pressure up to 25,000 because the lipid concentrations we were working with. So again, we can see before we use a homogenizer, they're very large and heterogeneous. After one pass, the size comes down quickly. 2 passes and 3 passes, we can just refine the product to that respect, to bringing our PDI down. Again, monodispersed population. Zeta potential is negative now because there's DSPG in it, a negatively charged lipid. Drug loading, very high, 100%. And here's some vesicles here, and they're generally one bilayer, but you get the odd one, which is a double bilayer. That's why we put that in here. But the size is monodispersed. So we also sterilize them so they were good to go if they were to be used as a product. Size is good, 100 nanometers, PDI 0.2, negative zeta potential, as we would expect, and very high drug loading. Basically, what you can see through those 3 case studies are we can make a range of lipid-based nanoparticles, and they can be produced in a scalable, solvent-free manner. So we have also looked at solid lipid nanoparticles as well, but I didn't cover that today. And it's scale independent, so getting us from the bench into manufacture in that respect. So it's very exciting times, and the fact we don't need to use solvent opens a lot of doors for manufacturing processes.